Many types of plants respond quickly to external stimuli. Here we see the leaves of a Mimosa Pudica close in response to being touched. When we repeat the movie, note how the leaves close one after the other in a series. The compound leaf of a mimosa is held erect by special groups of load-bearing cells that act like hinges. These groups of cells are located at the junction between the stem and the compound leaf, at the bases of the individual pinnae, and also at the bases of the tiny leaflets. When stimulated by touch, cells in the leaflets generate an action potential that propagates along cells. The signal causes the water pressure within the hinge-like cells to drop rapidly, and the leaflets begin to close. While this collapse can happen in several seconds, it takes several minutes for the water pressure to build again inside the cells and return the plant to an upright state. Note how a single strong bump can cause dramatic responses at multiple hinge sites throughout the plant. The process begins when mechanosensitive ion channels in cells of the leaflet are activated. Although the exact mechanism is unknown, it is likely to occur either through linkages between the plant cell wall and the ion channel, or simply by stretching of the plasma membrane. In either case, opening of the channel causes an influx of ions that depolarizes the plasma membrane and initiates an action potential that propagates along the membrane. Since the membranes of plant cells are interconnected through structures called plasmodesmata, which are the plant cell equivalent of gap junctions, the action potential can pass from cell to cell, down the length of the leaflet, to the cells in the hinge region. When the action potential reaches a cell in the hinge region, membrane depolarization causes an influx of calcium ions into the cell. This causes a large number of chloride ions to leave the cell, followed by an efflux of potassium ions. This mass efflux of ions causes water to rush out of the cell through aquaporins, which are abundant in these cells. The water pressure required to keep the plant organs erect subsequently collapses, along with the supported structures. As water returns to the cells, the plant will return to its former state. After a mimosa collapses and regains its form, it is often desensitized for periods of time, as we see here. It is speculated that the remarkable movements of the mimosa evolved as a defense mechanism against herbivores.